Well, it's a big privilege to speak in front of the most talented Israeli students.
Could you speak
Okay.
to the
Okay, so I'm going to show you, to demonstrate today how you can use tools in computational and quantum chemistry to study a basic physiological process such as division. So my talk is, the outline of my talk is the following. So I'll have first a quick introduction about the process in vision and about the proteins that play a key role in this process. Then I introduce the methodology that we use to study this process. And then I have two sections of results, two aspects of the process in vision. First, the process that is triggered by light. So it's the photo-induced process. And then I will talk about the thermal process, which determines the limit of our vision at night. So let's start with an eye. The reason why you can see this presentation and me is because our eye, shown here, focuses the incoming light through the lens on the backside. The backside is called the so-called retina, and it has two types of cells. If we zoom in into the retina, we'll see we have, on the one hand, rod cells, which have this elongated shape, and we have cone cells. So now, in one eye, we have 90 million rod cells, and we have 4.5 million cone cells. And they come in 3 different flavors. So the cone cells absorb blue, green, and red light. This is so-called absorption spectrum. While the photopigment in the rod cells absorb light at 498 nanometers. So the rod cells are responsible for dim light vision, which is the vision at low light conditions, while the cone cells determine our color perception. Now, the research that I'm going to show you now is focused entirely on the pigment in the rod cells. So here you see, this is a scheme of a rod cell. It has this elongated structure which resembles an antenna. And in the outer segment of the rod cell, we have a high concentration of so-called disks. And if we zoom in into the structure of a disk, we'll find a transmembrane protein. It means there is a protein which is integrated in the membrane of this disk. It has 7 helices, as shown here below, which goes through the membrane, and they form sort of barrel-type structure. So inside the barrel, in the middle, shown here in red, there is an organic molecule which is just half of vitamin A. And this molecule is called retinal. In rhodopsin, it's present in the following configuration. So it has 6 double bonds. All of them are in the trans configuration, while the one in the middle is in cis. So since cis and trans describe a configuration of a double bond, it's a nomenclature to describe whether the substituents are on the same side of the double bond or on opposite sides. So now, the primary event in vision, how everything starts, is when retinal, which is also called a chromophore because it absorbs light, it's sensitive to light, absorbs a photon of the wavelength 500 nanometers, and then undergoes a conformational change, which is called in chemistry isomerization. It goes from cis, shown here, to trans. And this is one of the most efficient processes in nature. The reaction is completed in 200 femtoseconds. Who knows what is a femtosecond? Except Barak, she probably knows. 15, yeah, exactly. So it's 10 to the minus 15. It has a 67% quantum yield. It's one of the most efficient photochemical energy converter. And it has 100% bond selectivity. Meaning, from these 6 double bonds, only one, the central double bond, is isomerizing. So this is a dream reaction for organic chemists. They would like to have such a selectivity in the lab. Now the question is, how does the protein optimize this reaction? Because if you take the same molecule, you take it out of the protein and put it in a solution, in ethanol or methanol, all these properties are gone. The reaction time is on picosecond timescale, so it's more than 3 orders of magnitude slower. The quantum yield is below 20%, And instead of one specific isomer, instead of one all-trans retinol, we have a mixture of different isomers because different double bonds isomerize. Okay, so one question is, how does protein control and tune this reaction? And the second question is, how does retinol isomerize in the very tight protein pocket? Because there's not much space. So I have studied this process, so-called isomerization mechanism, using computational methods. And if you go to an organic chemist, like Angelica, and you ask, how does isomerization occur? What is the mechanism? She will probably say it's a classical one-bond flip, meaning that you have two ends of the molecule, and one is rotating by 180 degrees to form the other isomer. But in a very tight protein pocket, there is not enough space to make such a large motion. It's not sufficient space. So, so this definition in the IUPAC Golden Book of the one-bond flip mechanism is not valid in protein. Now, it's quite interesting to know that even before I was born, people thought about it. Aryeh Warshel in 1976, it's really pioneering work because at that time, in 1976, there were no computers. So he got access to the supercomputer center of NASA to do these pioneering simulations. There were no commercial quantum chemistry packages like they're available today, so he wrote his own code. He got this computer time, and there was no protein structure. So he was just making several assumptions. Nevertheless, he came up with a mechanism where two double bonds isomerize at the same time. So two double bonds make a rotation which resemble the motion of bicycle pedals. And that's why he coined it the bicycle pedal mechanism. Well, as I said, he made several assumptions by fixing the ends of retinol because the protein structure was not known. He used van der Waals spheres to model the environment. And yeah, it was, at that time, it was the only mechanism that could explain the ultra-fast and the efficient isomerization. Then a few years later, Robert Liu, who is based on Hawaii, probably inspired by the local dances, he came up with the Hula twist mechanism. So he said, instead of two double bonds, there is a double bond and the adjacent, the neighboring single bonds, which do this isomerization at the same time. And he proposed this isomerization. So this is the double bond, this is the single bond, and this is the product. He said that the Hula twist mechanism requires even less space than the bicycle pedal, and that's why it should be preferred. However, there is a little tiny problem. It doesn't match with the experiment. So it didn't produce the all trans, but it gave the 10 S cis, meaning the single bond is in the cis configuration. Okay, and here at the end I show you the original image from the publication, where he represents a molecule as a dancing hula twist. So a few years later, the group of Massimo Levucci from Italy came up with a crankshaft mechanism. So they claim that instead of two double bonds, there is a whole series along the whole molecule, the so-called molecule backbone. There is a simultaneous twist of several double bonds. And because there are several double bonds, it's enough to twist them just by a little bit. It gives an overall rotation. Okay? So then after 2007, a group from Hans Lischka came up with the folding table mechanism. They say that there are three double bonds. The one in the middle rotates in one sense, and their outer double bonds rotate in the opposite sense. And by this orientation, they will reduce the space. So, and if you think this is funny, wait until the next slide. The group from Ishida came up with the ring a wet towel motion, where they say, like, if you want to wring a towel to get the water out, you just highly twist it, and it resembles slightly the crankshaft, but all the double bonds are rotated in the same sense. Okay, so I hope the motivation now is clear why we wanted to look into it. And I have to say, in addition, that rhodopsins are not only important in vision, they also play a key role in other biological and physiological processes. For example, a relative of rhodopsin, called melanopsin, is responsible for our sleep and awake rhythm. So it's a similar receptor which sits in our brain, and it's responsive to blue light. So that means if you play with your phone at night, you stimulate melanopsin, which gets excited and then perturb your sleeping cycle. So that's why it's harder to fall asleep if you play with your phone. So I recommend you to use a blue light filter. In Drosophilas, rhodopsins are responsible for thermal sensing and hearing, and the most recent field is using channel rhodopsin, which are used to regulate living organisms by light. So the channel rhodopsin is expressed in a neural cell, and then this neural cell becomes sensitive to light. And there is a very famous experiment where a mouse infected with this channel rhodopsin is switched with light on and off. So it starts to run when you have light. In absence of light, it just stops. Okay, so if you feel, I hope you feel inspired by my presentation, and when you get home, you go to the protein database. You can download the rhodopsin structure, and you immediately realize, oops, it has 5,521 atoms. So such a calculation on quantum chemical level is nearly impossible, even today, with the best computer. So lucky enough for us, these three gentlemen came up with the so-called multi-scale model or multi-scale approach, which allows you to treat such large system at very high accuracy without a big trade-off. And the way how it works is demonstrated here. So this is a photograph of Alfred Nobel. And now the photograph is divided in 3 different zones. In order to recognize the person, we need to focus on the face, right? So the face, we use the face to recognize the person. Now we have the highest resolution, the first zone on the face, and we recognize this is Alfred Nobel and not Theodor Herzl. So here we have the highest resolution zone, and if we move out of this zone, you see the picture is getting pixelated. So we are losing resolution, but we can also say that we like compress the information, like we heard in the first talk in the morning. And then if we move out of the second circle here to the third zone, everything is just getting smeared and blurry. So we lose completely all the detailed information. Now the same concept can be transferred to an enzyme or a photoreceptor protein. Because in the active site, in the center, or where we have the chromophore, we have the highest resolution. The highest resolution means we have electrons and nuclei. Now if we move out of the central of this active site, we have the protein environment. Here we reduce the resolution. We don't have electrons anymore explicitly. They are included in the parameterized force field. So this makes the calculation of the protein extremely fast. Then once we move out from this protein to the environment, to the solvent or the membrane, we can even model it with a dielectric constant, which would correspond to this gray blur mix. So we lose completely the resolution. There is no nuclei, no electrons. Now, this method is also called hybrid quantum mechanics, molecular mechanics, because the region of interest, in this case the retinal, is described using very accurate quantum mechanics, and the surrounding environment, which does not actively participate in the rearrangement of electrons, is described using molecular mechanics or classical mechanics. Now there is one little detail that I have to mention, that actually there is a covalent bond, there is a chemical bond between the chromophore here and the protein. So we also, once we make a boundary between these two regions, we have also to cut a bond. And once we cut a bond, the molecule gets completely different properties. So there is a little price to pay, and we have to parameterize this boundary and use specific so-called link atom. But nowadays this is solved. It's not a big issue. Okay, so now this is going to be the method that we use to take the picture, okay, to stay in this analogy with the photograph of Alfred Nobel. But we are not interested in the photo. We want to make a movie. So how do we make a movie? We run molecular dynamic simulations. And here I have one slide to explain how molecular dynamics works, because in chemistry or also in biology, molecular dynamics is usually associated with a classical force field, but it doesn't necessarily have to be a classical force field. So in molecular dynamics, you start at the point on the potential energy surface. So it's like a map. You have here the coordinates, you know the time is time 0, and you have the velocities. And what you do is, with the given coordinates, you can use any method to get the energy here, potential energy V. You can use the wave function method, classical force field, Monte Carlo, stochastic, whatever you want. Then you calculate the derivative of the potential energy, which is just the negative of the force. And you use the force to get the acceleration. And then you can use this acceleration to plug it into Newton's equation of motions. And then you get the velocities for the next step. So meaning, by doing these calculations here, we get from time t0 to the next point, where we have the new coordinates, new velocities, and new time. And then we repeat the cycle again, and we go to the next point. Okay, I hope this is clear.
Does the potential change as a result of the motion?
Yeah, the potential is different at every point.
No.
In time.
Yeah, does the potential changes in time as a result of the motion? Or is it static?
No. This, I have to say, this is the potential energy, and it changes in time, yes. Okay, so maybe if it's not clear, I prepared a little animation to illustrate the concept. So you can imagine now it's the season for skiing. So this is a mountain. You're somewhere on the top, and you progress from point T to T1 to T2, and so on. So basically, you have some coordinates at the beginning. Then you solve Newton's equation of motion. You go to the next point, and so on. And at the end, you can just combine these images and make a movie.
So this isn't a good analogy, because here the mountain is static. It's not affected by the sphere.
The potential energy is different at every point of the mountain.
Yeah, but it's not time dependent.
So you move along a static potential, but in reality, sometimes the potential can also change in time.
I think you need to distinguish between the one thing is potential and the other thing is the potential energy. So in molecular dynamics, we have both potential energy and kinetic energy. This together gives you the total energy. And so using kinetic energy, we propagate from one point to another. And at each point, the potential energy is different.
Each point in space, not in time, right? It's fixed for all times in the case that you don't have an oscillating field or something like that. That's what
Imagine another molecule comes and then.
Let's say the molecule also evolves, not only the.
Yeah, the molecule does evolve. The molecule, it's not okay. It's not a fixed molecule that moves. This is it's just we have 3n minus 6 degrees of freedom, and is a number of atoms. So it's hyper-dimensional surface that I mean in a simplified way.
Yes.
Okay.
No, so it does. It's.
I will show you the movie now, and then I hope it will become more clear. So all these methodologies, this QMMM and the molecular dynamics, has been implemented by me into the Molcast program package. I don't want to go into detail, but just to mention that this program is available for free. You can go and download it, and in the latest version 8. And you can, if you want, you can run your own simulation on your laptop. So with this, I will just go to the results. So this is what we get out from our simulations. So for clarity, I removed the protein in this visualization. Here you see only the retinol. This is the conformation. So these are the different bonds. And in addition, I'm also showing you the electron density. So this is the electron density in the pi orbitals. You remember we had 6 double bonds, and you can clearly see wherever you have this red density, it shows you the position of the double bond. So the double bond has a symmetry. You have a pi cloud below and above the plane. So this is clearly a double bond. Whenever we have a knot in the density, this is a single bond. Again, we have a double bond, single bond, double bond, single bond. So now this is the situation before a photon is absorbed. Now, when I start the movie, in this very moment, we have an absorption of a photon, which is an extremely fast process. And then you will see that first, upon absorption, the electron structure is changing. Now, the double bonds become single bonds. Here's a knot, and these single bonds become double bonds. And then once this change is completed, we have a rotation. And you see these two hydrogens are now on opposite sides. So let me show you again. When we start, both hydrogens are on the same side. So we have the cis configuration. Now we're going to excite the molecule. It undergoes the double bond breaking. Then we have these hydrogens, they move out of plane because now there is a knot, so they can freely rotate. And once this rotation is completed, the double bond is restored in the middle, and you see these hydrogens now are on opposite sides. So we have simulated 200 femtoseconds, and we have basically observed a photoisomerization process. And coming back to the mechanism, we see here so-called bicycle pedal mechanism, but instead of two double bonds, only one is fully isomerized. It means only one goes from cis to trans. The other one is highly twisted. And that's why instead of coming up with a new name, we decided to call it an aborted bicycle pedal. So, but we can go beyond this, molecular dynamics and just the simulation of the dynamics. We can also predict the stereoselectivity, which basically says how the protein and the environment controls whether we reach the product in trans or if we go back to the cis. So at a 90 degrees, so now we are looking along a double bond, and in this situation we have a twist of 90 degrees. So the p orbital here in dark gray and the p orbital behind in light gray are completely decoupled. So in order to have an overlap between p orbitals, they need to be aligned. Now this is the worst possible situation for p orbitals. And now from this point at 90 degrees, there are two possible scenarios. The hydrogen atoms can move towards each other. Okay? This will increase the overlap of B and B prime lobe, and this will lead to the trans isomer. While in the other case, if they move towards each other, it will increase the overlap of B and A prime, as well as B prime and A, and this will go back to the cis. Now, I told you at the beginning, the ratio, the quantum yield is around 65, 67%. So it means in nature, 65% completes the reaction and 35% goes back. And when we came up with this mechanism, I could not imagine that it's possible to assess or influence this reaction which takes place in 100 femtoseconds. But there is a group in Berkeley that came up with a very clever experiment. They have substituted hydrogen with deuterium. So deuterium has twice the mass of hydrogen, and they have found 3 different isotopomers, so 3 different patterns. They replace both hydrogens with deuterium. This is one isotopomer, and in the second and third, they replace either one of them. And what they found is if both hydrogens are replaced with deuterium, then the ratio remains the same, because both hydrogens are replaced with equally heavy atoms, and the symmetry is maintained of this hydrogen out of plane mode. While if you replace only one of them, it becomes asymmetric, and then they observed 55% of the cis and 45% on trans. So indeed, it proves that our understanding of the stereoselectivity was correct, and actually, I think it's one of the examples where the theory or simulations were leading the way for experiments. Okay, with this, I want to close the first part. Maybe if you have some questions before I go to the second part, you can, yeah? Sorry? Yeah.
You said that there were a series of changes, a series of zones, sequence zones.
Yeah. Well, I mentioned this when I was discussing other mechanisms proposed by. So what happens when the photon is absorbed, then the electronic structure is changing almost instantaneously. You remember this red densities that they have? the electrons are much faster than nuclei. They immediately change upon photon absorption because the molecule is promoted to an excited state. And then the nuclei, which are much heavier, it takes them a few femtoseconds to adapt to the new electron configuration. So then as a consequence of a new electronic structure, the nuclei start to move slowly. And from what we have simulated, we see that the motion is such that one double bond is fully rotating, while the second one is partially twisting. And this allows to isomerize in very small space.
Yeah, you probably said it and I missed it, but why doesn't it happen actually in vitro? Why doesn't it happen only in cells? Why is it less efficient in testing?
Okay, you mean in solution? Okay.
Yeah.
Well, there are a few reasons for this. So, in the protein environment, I haven't mentioned this, but here, so if you look at the structure of retinol, the link of the retinol to the protein is called the shift base link, and it has a positive charge. And in the protein, there is at this specific position, there is a carboxylase, which has a negative charge, and it's kind of counterion to this positive charge. So there is some electrostatic tuning from the protein, which is not there in solvent. And also, the protein environment has a steric effect, which it confines the space. So it just it doesn't allow any other rotations and forms. In solvent, this is not the case. So there is the steric reason and also the electrostatic reason because of the missing counterion. Any more questions? Okay. So and then I go to the second part, and so far I told you about the process where we have a photon, which is absorbed by our molecule, and then as a consequence we can see an electric signal. So this is a measurement by the Yao group from Yale University. They can extract one single rod cell and measure the electric response in picoampere. So when they do these measurements, you see there are some peaks which indicate an electric signal coming out from a rod cell. Now, when they switch off the light, in total absence of light, there is a very rare signal which is exactly of the same amplitude, like a signal triggered by light. So the question arises, how is it possible that the molecule undergoes the same change without any light? And the only explanation they came up with is that instead of the photochemical isomerization, there is a thermal isomerization. And what they also measured is that the signal coming from the thermal isomerization is completely the same, is identical to the photoactivated. So there is only one little problem. The barrier for thermal isomerization is so high that we could observe only one reaction in 400 years. But if you remember at the beginning I told you, in one eye we have 90 million rod cells. This is a rod cell. And each rod cell has 2 billion rhodopsins. Because of this high number, even such a rare event becomes probable. So this rare event produces noise because we don't actually see anything. There is no signal, but we still produce an electric signal. And this was first observed by German scientists who coined this Eigengrau, which means intrinsic gray. And this is responsible for the uniform dark gray background that many people report seeing in absence of light. So if you close your eyes at night, at the beginning it's not, you see some white dots, and it's not completely black, but kind of bluish, grayish. The reason for this is the thermal isomerization. So in order to probe this, Sir Horace Barlow in Cambridge, he extracted rod cells from different organisms. And he observed a very interesting trend. So he noticed that the bluer the absorption, so the absorption of the photon is going to the blue side of the spectrum, there is a correlation with the blue absorption, with the blue shift, and the low thermal noise. So the bluer the absorption of the pigment, the lower the thermal noise. And the opposite, the more it shifts to the red, the higher the thermal noise. So this was known since 1957. However, this physiological phenomenon was lacking in mechanistic underpinning. So although it was known, there was no understanding why there is a link between this light activated and the dark process. And we have used exactly the same QMMM model to explain this so-called Barlow correlation. So before I show you the result and I give you the explanation, I just want to summarize these two different processes. So on one hand, we have the photo process that I explained you in the first part. So this is the so-called electronic ground state in blue, and this is the red. In red is the excited state, and you can reach, the molecule can reach the excited state by absorbing a photon. So this is indicated by a vertical line. Now, this excited state is characterized by a different charge distribution. You see here blue spheres and red spheres. Blue is positive, red is negative. Now, when you excite the molecule, you see these spheres are changing, they are rearranging. So in the excited state, we have a different charge distribution. Then, this photon has a characteristic lambda max, which basically describes the excitation energy. And once we reach the excited state, the molecule is propagating through a topological feature called the conical intersection. I will explain in the next slide what it means. And then it relaxes back to the ground state and forms a trance. So this conical intersection is like a funnel. So once you get inside, you immediately go tunnel through and go to the ground state. Now, in contrast to this photo process, the thermal process occurs entirely on the ground state. And the barrier is characterized by a so-called transition state. Transition state is roughly in the middle between the reactant and the product, and it's characterized by the thermal activation energy shown here as the barrier. So we need to overcome this barrier in the ground state to reach the product, while in the excited state there is no barrier. We just absorb a photon, the molecule evolves through a conical intersection, and reach the product. Now, basically, the Barlow correlation is a correlation between this energy here, the height of the barrier, and the excitation energy, the energy between the ground and excited state. So now I will explain you what's the connection between them. But before, I will just briefly explain to you what is a transition state and what is a conical intersection. So when we go in chemical process, if we start with the reactant A and we reach the product B, we usually go through a transition state, which is characterized, it is a stationary point on the potential energy surface, and it's characterized by a transition vector which describes the direction from A to B. And it allows you to reach one product. Now, in case of the conical intersection, it describes as singularity. So this point cannot be optimized, and it has two vectors. Okay? If we follow one vector, we reach the product, while in case of the other one, we go back to the reactant. And this conical intersection allows very fast femtosecond reactions.
Take a microphone.
Okay. Okay, so this conical intersection allows the reaction to complete it on a scale of femtoseconds, and like a funnel, it allows the molecule to relax back to the ground state. Okay, now if we want to simulate these two processes, we need to have the reactant and the product, and then they can be connected with each other in two different ways. Because of the presence of the conical intersection, and here this is just the lower cone, we can move on one side of the conical intersection to reach the product, or we can move on the other side. And there is a consequence of these two pathways. So we have now, instead of one, we have two transition states. And according to the geometric phase theorem, or Herzberg-Loget-Higgins theorem, if we displace the molecule around the conical intersection, the wave function describing the molecule changes its phase. So in simple words, we have a transition state which has one character, here at the radical, and here in this brown portion of the potential energy, it has a charge transfer character. Meaning, we can break the double bond in two ways. One, you see here these two dots. This is a diradical, so we cut it in the middle, the double bond. In the other case, we break it by putting the electron pair entirely on one side of it. So it means we have two different transition states, and they have different energies. And we found out by calculations that the charge transfer, if you remember, it's like the excited state, the charge transfer, is 11 kcal per mole lower than the radical transition state. And now we can explain the Barlow correlation. So here I'm showing you the excited state, this is the ground state. So now, okay, I forgot to mention one important thing that is maybe obvious for biologists. But by comparing rod cells from different organisms, the only thing that has changed is the protein environment. So the retinol is always the same, the conformation is always the same, but some of the amino acids, which are the building blocks of protein, they have been altered in different organisms. This is so-called mutation. So now the retinol is always the same, but the protein is changing, and this alters the spectroscopic, the excitation energy and the thermal energy. So now we simulate, for example, if we introduce now a mutant which has a negative charge, this one is going to stabilize the charge transfer state, you saw that, more than this ground state. And this will at the same time decrease the excitation energy as well as the transition state. So to prove that, we have simulated this Barlow correlation. So we plotted the excitation energy against charge transfer transition state and the radical, shown here. So basically, only the charge transfer transition state gives you exactly the same slope like the experimental, while the radical gives you the opposite slope. It gives you this slope shown here in blue. And we can also go beyond the experimental Barlow correlation. We can also predict that at some point, there will be a crossing between this red line and the blue line. It means if you shift the pigment very far to the blue, which has not been achieved yet experimentally, then the radical transition state will become lower than the charge transfer. And then we'll have again higher thermal noise. Okay, with this, I would like to finish my talk and just mention my groups, which is located at the so-called Fritz Haber Center. So it's a center for theoretical and computational chemistry with 10 groups. And I would like to acknowledge my group members. Without their hard work, these results would not be possible. And thank you for your attention.
So for the sake of history, maybe I should tell the story, maybe you know it better than me, of the 3 Nobel laureates here that won Nobel laureates in chemistry. So this Nobel Prize actually should have belonged to Israel, to the Weizmann Institute. The scientific father of all 3 of them was Shneur Rhodopsin at Weizmann, and he raised a generation of very good people. So Aryeh Warshal is a kibbutznik, was a former kibbutznik who did his PhD there. I think Michael Levit, by the way, Warshal did his bachelor and master's at Technion, then moved to Weizmann to work with Rhodopsin. I think Levit came as a postdoc, if I remember.
From UK, yeah.
Yeah.
He grew up in Britain.
In Australia. I'm not sure.
I think he's from
Karplas came as a postdoc. All 3 of them worked for Shneur Rhodopsin. So he deserved the Nobel Prize. He died in 1992 or something like this. So coming to look at the 3 of them, so 2 of them wanted to stay at Weizmann. Karplas left after the postdoc for a position in the US. And now he shares his time between the US and I think
Strasbourg.
Warshal and Levit, one of them did not get tenure because Weizmann said that they have And after crazy guy, one crazy guy, Shneur Lipson, that is doing this kind of strange stuff. So Marshall understood that his chances of getting tenure are small, and he left. Levit is married to an Israeli wife, so he wanted to be part-time. He said no, so both of them are in the US. So this is how Israel missed 3 Nobel Prizes there. One of them because the father of all this idea died, Shneur Lipson, and the other 2 because some of the leaders in that institute were stupid. Unfortunately, they were, I'm not saying that the Technion is not doing similar stupidities, but this is how we missed very good people. And now they are friends and they are coming. I think Levit has part-time appointment at Weizmann even now. He has he lives there half the year. But still, it would have been much nicer that they've been here. So let's entertain a few questions to Igor for the fantastic talk. So let me go back to Warshaw's prediction. Okay. So what you said is practically that on that prediction, that pioneering explanation is incorrect, practically speaking. The second one, the one on the, how do you call that, bicep, is partially correct. But
yeah, actually, no, it's actually Warshaw's mechanism is almost correct.
I see.
So
I thought that the second one is, is partially correct, but Warshaw's
yeah, actually, yeah, it's actually, I have to say that at that time in 1976, experimentalists did not believe that an isomerization, this rotation, can take place on femtosecond timescale. So they assumed that the primary event in vision is a proton transfer. So in this sense, I think it was really a brave prediction that he, computational chemistry is even today hardly recognized by some of my colleagues, but he did it in 1976, and he was brave enough to propose the correct reaction and also the almost correct mechanism. So I think it's really outstanding. And this was just one of the, so this was
made many,
an application, and then he also developed the methodology in the same year.
They are using now in pharmacology, these methods are being used widely. You know that physicists like myself, usually we look down on these kind of things. We say, this is not real quantum mechanics. This is like Play-Doh, plastilina. You play with it and you put H-bar and you decorate it. It's only fancy, hunky-punky, and it never works, and there is no predictive power. But of course, now, since 90-something, the physicists were proven wrong. Actually, they were right. And sometimes it's important to acknowledge that the beauty is in the approximations. Because we cannot compute everything. And we can, and sometimes you do some approximations that can lead to illumination.
Yeah, I think it's due to advance both in development of algorithms and approximations, but also the computer industry underwent a really fast development. So
if all will be okay, maybe in 10 years you'll have a quantum computer that can do real computation of this, and then you will find that you are right.
Okay,
Thank you, Igor. Thank you.